Hey everybody, we're going to be taking a look at chapter 13 this week. So we're skipping around just a little bit. We go from chapter 6 about viruses and we jump all the way to human microbe interactions. So we'll come back a little bit. We'll actually end up going back to the middle chapters here and end up looking at chapter 7, 8, and 9. So for now, I'm just going to go ahead and look at this topic, human microbe interactions. So chapter 13 gets into some pretty interesting things. We'll try and keep it to the most straightforward, and I'll try and bring in some new outside information to supplement this. But as I've been mentioning, and as you've been reading about in some of the online articles, that the whole field of microbiology is seeing a kind of a surge with new research and new indications of the relationships that humans and microbes have. So I'm going to touch on that a little bit and give some examples of some of the most current research within the last year and a half, give or take. So starting out with what we kind of already know, what I've already mentioned, what you probably knew coming into this class to a certain extent, is the human body is host to trillions of microorganisms. They exist in a combination of mutualistic, commensal, and even pathogenic symbiotic relationships. Remember back in chapter 1, I defined these types of relationships and talked about how these relate to people and humans and other animals. To clarify, most of these are bacteria, the vast majority. So we're talking a very, very dominant bacterial microbiota. But we also actually have fungi as a fairly common type of microbiota, and even protozoa, although less is known about that and certainly fewer types of protozoa. But it is known that those are typically part of the normal human flora or normal biota, and even viruses. Now that part is even more speculative and even less well known. But viruses are as ubiquitous as any microbe. And they find a lot of actually bacteriophage viruses that infect bacteria in the human body. So whether they infect people or not is another question, but they are naturally part of the flora, especially those that infect bacteria. Generally speaking, we call these the normal biota. Normal biota is a generic term for all these microorganisms as they exist in the human body. There are other terms such as normal flora, microflora, but generally speaking, we'll refer to them as the normal biota here. As we've already talked about, and as this bullet point indicates, been finding newer research indicating that the full extent to which they affect our physiology and even psychology. Remember the online article about microbes and how they affect our mood from the New York Times, one of the first articles I had you read. So what I find so fascinating about this particular field, microbiology, is that we've been studying it for almost 400 years now. I mean, microbiology really goes back to the discovery of the microscope, or I should say the invention of the microscope. And the very first was called a single-lens microscope, invented by a man named Antoine van Leeuwenhoek. And he was the first person in the 1600s to see these microbes, and since then, the whole field of microbiology has just been growing gradually. And you'd think that after 400 years, scientists and microbiologists would have a very, very fundamental understanding of all these microbes. But the truth is, as scientists do more research with new technology, they're finding out that we're really very, very limited in our full extent of what we know about these microbes. And it's very, very shocking as to the new research that kind of indicates that. So, for example, this textbook, as it appears, has some outdated information, and this is an example of what I'm talking about. The textbook reports this, and I've been teaching this for the last so many years. I'm actually just now updating this as I learn about this. But we've been talking about how in the human body there's an average ratio of 10 microbial cells for every one human cell. That's what we've been telling people. That's what it's been told to me and everyone else. And the indication then is that your body is more microbial than it is human. By a factor of 10. So that you actually, the theory on this was that you have 10 times more microbial cells than you have human cells. But now they're saying that this is probably most likely way, way off. And the most current research that we have to assume, I guess anyway, is more accurate, has really kind of demystified this whole ratio, which always seemed pretty extreme to most people. now they're saying that that's not accurate at all. So this is a fairly basic thing to establish how many cells we have, how many microbial cells we have in the human body. It's a fairly basic thing in terms of what the full extent of research you could be doing. It's basically a counting exercise, an advanced counting exercise. But to reiterate my point is that only in 2016, January 2016, is a more accurate count of microbes even now being established. So to clarify, what they're now estimating is that it's probably closer to 1-to-1. In the latest study I was reading put this at actually about 1.3 to 1, meaning that we probably have about 1.3 microbial cells to every one human cell, but really pretty close to 1-to-1. So when we get to the review questions and when it comes to questions about this ratio, I'm going to ask you what's the old ratio and then what's the new ratio. So know that the old ratio, the outdated, was 10-to-1. In the new ratio, I'm just going to keep it at 1-to-1. I'm not going to try and trick you with 1.3 or 1.6 or anything like that. This 1.3 is just a little bit more of an accurate estimate, but they're basically saying it's actually about 1-to-1, about one bacterial cell for one human cell. And the truth is, microbes tend to exist in certain body locations very, very heavily, and then tend to be fairly sparse in other areas. So, for example, most microbes are actually in the gut, and probably much fewer in say, the respiratory tract or in the even in the small intestine as opposed to the large intestine. But to clarify, the gut or the colon is the large intestine where most of these microbes are. So many microbiologists have been quoted as saying that it's estimated we are currently in our quote-unquote infantile stages of our full understanding of microbes. And that's simply just an analogy to kind of put into perspective how far we have yet to go. So the idea is we're comparing it to the development, say, of a child from the very, very prenatal stages all the way up to an adult. And right now we're in the infant stages. And what that really means is that we've probably got many decades yet to go before we have a really, really true fundamental understanding of what's actually going on here. And so as I was just saying earlier, this example of just trying to understand the number of cells in the body is an example to kind of indicate that point there, that we really have so much yet to know. And what's a little bit frustrating about this is that some of this information gets reported, and it's hard for everyone else who's just reading the information to know where it comes from. But as it turns out, this number 10-to-1 came from a really just one study done back in the 70s that people took and just repeated over and over, and that was never really meant to be a fundamental number of a concrete proportion. It was really more of a rough estimate based on very limited research that just for whatever reason got passed down and became part of the central information in microbiology. So it really kind of is frustrating to know that some of the key facts that I've been teaching and other people have been reporting are actually based on some fairly loose research. So part of it has to do with the reporting and the nature of research. Part of it has to do with our lack of knowledge. So there's a consequence of both here. But the idea is that we have so much yet to know. Here's a link to that study that I was talking about here. And what they're actually saying is that in the human body, the most dominant cells are red blood cells, more technically known as erythrocytes. They make up the vast majority of cells in the body. Another number that I've been hearing way over reported is that the human body has about 100 trillion cells. This is actually probably closer to about 37 to 40 trillion cells. And of those, then about 25, give or take, are typically red blood cells. They make up the most dominant cell type in the body, quantity-wise. In one of the review questions, we'll ask you, what's the most numerous cell? That's the red blood cells. Cells that are most numerous by mass, those that take up the largest amount of body weight, are actually our fat cells and our muscle cells. They make up, you see here in this kind of green and then the gray here, these represent a proportion by mass. So quantity, red blood cells, mass, fat, and muscle. So red blood cells just don't have much mass. There's not much to them. So there's lots of them, but each one are so small that they barely take up much space. So those are actually the most common cells in the body and actually take up the most mass. And when they've been doing these estimates, one of the things they were doing was simply taking a small portion of the human intestine, counting the microbes in that, and then scaling that up and saying, well, if there's 1 million microbes in, say, a gram of body weight or gram of body tissue, then there must be, that number throughout the whole body. And what they're saying now is that that's just not true. There's high concentrations in one area and then very, very low concentrations in other areas. And so that type of quantifying really is just not accurate, and it doesn't really give us a good detail on what's actually happening there. So those are some numbers about what's actually happening in the body here. So going on, when we look at microbes, we see that the body really is not only just a source of nourishment for microbes, but a vast array of different nutrient types for microbes. So in other words, you can really think of the human body is like a different series of selective and differential media, probably more selective media for different microbes. So think about in the lab when I talked about selective media and how media can be tailored to grow one microbe but not another, and this has to be based on the types of nutrients and the ingredients that we put in the media. Some are toxic, some are selective in that sense. You can really think of the human body in a very similar way, just from a more natural standpoint. We have all these different body locations, and each one has a different pH, temperature, oxygen level, nutrient content, and each one of these factors will affect the range of microbes found there, much like a selective media would in the lab. So it's a good way to think about the human body. So for this reason, then, some microbes are well suited to the skin, like Staphylococci. Some microbes are well suited to the intestines. E. coli, a lot of gram-negative microbes tend to thrive there. And a variety then establish themselves in the respiratory tract, gram-negatives, both gram-positives. So each part of the body can be thought of as a kind of a selective media, and this is because of the unique environmental conditions here. So another question in the review is, why are microbes established in certain areas? What factors influence the diversity? And this has to do with the different conditions, such as pH, temperature, oxygen, etc. One of the big revelations in microbiology, going back to roughly 2009, and really even before that actually, is called the Human Microbiome Project, and I'm going to ask you about this as well. The Human Microbiome Project is a research project established by the NIH, part of the National Institute of Health, government-funded research. And one of the things they set out to do was to specifically try and sequence and analyze, I should say culture and even sequence the genetics of microbes in the human body that were previously unknown. So around this time, it was becoming more and more obvious that the human body had all these unknown microbes, and this goes back to the inability to culture most of these. Recall that only about 1%, give or take, of all microbes can actually be cultured. So knowing all this, they decided to set out and try and find out as much about them as they could, because they were realizing at that time how much they actually knew, how little they actually knew. So their basic goals and objectives, and this is a question on the review, what were the goals and objectives? The goals were to collect genetic sequences, determine what microbes were just there, and especially focusing on those that were unculturable, and trying to then establish just what microbes were there, how many different types, and get an idea of what the different nature of these microbes were. And then, the big goal then was to finally determine what role these microbes play in health and disease, which ones are good, which ones are bad, and kind of start to categorize these and catalog these in terms of their impact on human health and disease. So they're still very much in the thick of it, and little bits of information trickle out of it, and research is coming from all over the place. Not all research is centered around this project, but they're still reporting, and they're still giving lots of indications of some of the importance that these microbes are playing without necessarily defining exactly their results, because really what's happening is they're collecting data at this point, and they're going to be doing that for probably another good 10 plus years. So unfortunately it's going to be a while before we get a really, really strong indication of what they're learning. But what they've come out with so far is really compelling, and that is from some of the early research they've done. They looked at test subjects taking samples from the body and found right off the bat that the ratio of human genes to microbial genes in the body, in the human body, is highly disproportional. Now, perhaps this number will be refuted, such as the first number. It's hard to say whether these numbers are going to prove to be as accurate as they're stated, but this is probably much more accurate. To clarify, this is actually based on some fairly solid science. Gosh dang, I'm sorry, I keep clicking that. So I would not expect this to change dramatically, but that's always a possibility. What's been known since about 2003, they did the Human Genome Project. The Human Genome Project set out to sequence the human genome and determine how many different genes were in the human body. And they revealed about 23,000 protein encoding genes. That number might be a little bit higher, but give or take around 23,000 protein encoding genes, which is not that many. It was a surprisingly small number considering what the estimates were. Estimates were in the hundreds of thousands to even millions, and the actual number came out to be about 23,000. So what that means is our entire body is more or less dictated by about 23,000 basic genes. What they're finding about microbes, however, is that their contribution is closer to about 8 million in terms of how many genes, protein coding genes, they have. So microbes have similar genetics that we have and contribute about 360 times more genetic material than our own cells do. Now, how many of these genes are important in the human body? That's a hard question to answer and impossible to say directly. But the indication here is that if genetics play a really big role in the functionality of a living thing, then perhaps just the quantity of genes is an indicator of the significance of those genes. And if that's the case, then the microbial contribution to genetics is pretty significant in terms of just the quantity of genes that microbes have that can influence our body. So that may not make perfect sense. Unfortunately, you may not have a lot of genetic experience coming into this class, and so if you don't, that's okay. Just recognize that genes are really fundamental units of life. They contribute the characteristics to living organisms. They're like the blueprint for life. So quantity is not always the best indicator of what's happening, but it's a good starting point. So with the starting research here, they're indicating that the genetic contribution from our microbes could be as much as 360 times greater than our own. That mean, I should probably re-clarify this statement. It means that the genetic contribution could be about 360 times greater than our own. Doesn't mean that each one of these genes has an equal contribution. So there's a potential there for that much greater impact than our own cells. And this really goes right along with what all the other research is indicating. What we've been talking about is that microbes influence our body and that their presence has a major impact on health and disease. So this is a little bit more of a specific quantity of to how much they're affecting us, but goes right along with what we already know is that they do have an impact on the human body, and so this kind of puts that into more of a specific numerical context. So as an example of what some of these are thought to be doing here, some of these are enzymes that help us digest food, for example, and this is just one example of what these genes could be doing. But a certain number of these would actually be found in gut bacteria in the colon, and they can actually release some of these proteins to help break down food and to digest nutrients. And that's something that's actually been known for quite some time, that microbes actually help us digest our food and play a major role in our metabolism. So that's an example of what these genes might actually be contributing, probably a much greater extent, but that's a basic example of what they might be doing. Now, another interesting thing that the research has indicated is that where previously locations in the body were thought to be sterile, they're now finding unculturable microbes. So if we go back to the beginning of microbiology, what they were doing to study the body was much like what we would do in the lab, where they would take swab the body and just put it on a plate and then see what grew. And that kept them busy for decades because they'd always culture something. But as we now know, that culture rate represents probably only 1% of all the microbes there. So knowing this, these scientists set out to look at the unculturable microbes, and what they found is that these areas that were previously defined as being sterile because nothing would ever show up on the plates, on the petri dishes, what they're finding is that these actually have a fairly large number of unculturable microbes. So they're there, but they wouldn't show up on a petri dish. So that was another revelation of this Human Microbiome Project early on, is that our body really doesn't have very many sterile locations, not nearly as many as what were thought previously. And the other thing, and I mentioned this already back in chapter 1, is that all people so far studied harbor potentially dangerous pathogens in low numbers. So this goes back to what I was mentioning, is that the concept of a disease is changing. We've always looked at disease as kind of an infiltration, an invasion, so to speak, from the outside. We're healthy day 1, we have no bad microbes in our body day 1, and then day 2, we're invaded and from the outside, and those microbes establish and cause disease. But in reality, what they're really saying now is that microbes are probably already inside our body, and there's an imbalance that can happen that creates a higher number of dangerous pathogens. So we probably have a much greater impact by microbes already inside of our body as opposed to just those that would infect from an outside source. This here is some of the text, and it's listing some sites that have been known to harbor normal biota, and then sites that were thought to be sterile, and then some of these that are now being shown to no longer be sterile. Now, this hasn't been updated for a little bit. I haven't actually looked at some of the newest. I haven't actually seen any of the latest reports, but this is from the text, which was probably published in 2012, 2013, so a couple years old. But from the early culture studies, they found bacteria all over the skin, mucous membranes, etc. And now they're finding many other sites that were thought to be sterile, such as the liver, such as the blood, even heart circulatory system. They're finding unculturable microbes there that previously weren't being shown here. So along with this same line of thinking, then, what they're finding now is that people with certain diseases appear to have a different microbial makeup than people without some of these diseases. And so they're looking at links between specific types of microbes, either the presence or the absence, in relation to diseases such as autism, Crohn's disease, psoriasis. These actually are listed in the textbook. There's a number of other examples. And I pull up some research here, some recent research to show where they're at with some of these. And actually, let me just pull these up here real quick. One on Crohn's disease. This was published in, today, September 27th. I'm recording this September 27th, 2016, and this was published around the same time. So this is really recent. It's actually published today. So what they're saying here, new research is indicating that they think they found the cause for Crohn's disease. Now that may be a little bit of an overstatement. Sometimes they like to overstate their findings. I'm not saying that I know that to be true, but I'd be a little bit hesitant to say that they've found the cause indefinitely. But they found a link here studying 500,000 people, fairly large sample size, and what they found was that there was a connection amongst all of them, that they had a specific combination of different microbes. Specifically, what they found was that the microbes E. coli and Serratia marcescens, we work with both of these in the lab actually, in combination with the fungus Candida tropicalis, can produce a biofilm in the intestine that prompts inflammation and can trigger symptoms of Crohn's disease. So in this case, what they're saying is that it's actually a specific combination of 3 types of microbes in combination that form a biofilm that trigger the process of inflammation in the gut that leads to Crohn's disease. And if you're not familiar with Crohn's disease, it's basically a chronic inflammatory condition in the gut in which your gut becomes inflamed. You have stomach problems, pain, irritation, and so far they've not been able to do anything much about it. I think they can treat the symptoms and give medications. I'm not real familiar with what exactly they do to that, be honest, but I know that they've not had any sort of cure in people who have it, who just more or less just had to deal with it. Excuse me. They just had to deal with it, and now they've actually allegedly here found a link that perhaps they may be able to then go in and treat this with antibiotics or more specific protocols that would address the biofilm formation here. So that's fairly interesting. That's good news for anybody that has Crohn's disease. Even if they don't have a cure right away, that's a good indication that they're on the right track and that they may get to that point here relatively soon. Another interesting one, this is much more controversial, but the link between autism and gut bacteria. Now, autism is a spectrum disease. Autism, as you're probably familiar, is a neuropsychiatric developmental disorder in which mostly children and young adults have social disorders and sometimes learning disorders and a variety of kind of neurocognitive disorders. And while there's probably a lot of causes to it, one of the things that they're showing is that there could be a link here with gut bacteria. And that's not shocking because we know from other research that microbes in the gut can influence the human brain. That's been fairly well shown. The exact extent to which is still the big question, but the fact that they can affect the brain has been fairly well established. So this isn't necessarily a huge revelation if you understand how microbes can affect the brain. So they're showing here that there's some research indicating the nature of the gut microbes of women at the time of birth, and different species of gut bacteria that are present perhaps could affect the brain of the child during the developmental process. And interestingly, what they found was that the weight of the mother had an effect on the microbes in their gut. So what they've shown is that people who are obese have a different microbiota. They call it here microflora, by the way. We call it normal biota. Different terms for the same thing. But the microflora or the normal biota of people who are considered obese is different from people who are not obese or more of a normal weight. And what they're finding is that perhaps this difference in microbiota causes a link in the development of autism. So this is not quite as solidified a connection, but there's a number of studies like this that are finding similar types of links. So I would expect this to become more and more clear, and I would expect this to ultimately be shown to have a pretty significant link. But chances are there's other factors that go into this. It's probably not just microbes, but it may be. But I would expect there to be a number of factors contributing to autism. So don't take this as they're saying autism is exclusively caused by bacteria. That could be the case, but certainly at the very least what they're saying is that microbes such as bacteria influence that sort of characteristic, which is autism. And what's interesting is they go on to show that in mice, they did studies in mice, and they can find similar patterns of behavior in mice in terms of their social behavior. So mice can be asocial, can have autistic-like symptoms where they have social interaction disorders, and what they found was that this behavior could actually be influenced by gut bacteria. They could actually change the gut bacteria and actually influence the social behavior of mice. So while mice are very different than people, the idea is that we're similar in a lot of ways. So if the gut bacteria of mice can influence their behavior, perhaps the same thing could happen in people. So that's a couple of examples there. I have one more to show about psoriasis. Another one here. This one's a little bit older, going back a year and a half now. But another connection to bacteria, and this is actually looking at psoriatic arthritis, which is actually a condition that occurs along with psoriasis that can progress into arthritis. So psoriasis, if you're not familiar, I'll show you a quick psoriasis. Psoriasis is a skin condition that causes inflammation rashes and things like that, and people break out with these really nasty sores on their body, and this is really an inflammatory condition, considered like an autoimmune type condition, which the body reacts against itself. So basically you have the body attacking itself and causing these sorts of rashes and these reactions. And what they're thinking then is that this sort of self-attacking that the body does, called autoimmunity, can actually be triggered by different types of bacteria in the human skin. So all these are indications of the role that microbes are playing in the body. Okay, so let's go ahead and pause it here, take a look at the first set of review questions. These are a little bit more, I got a little bit more here. I'm actually going to only have 4 parts this time, so I ended up putting more questions into fewer parts. So take your time, and we'll go ahead and start the next part here in just a second. Okay, so adding to this a little bit here, normal biota in the body have been known to have a beneficial effect on humans, and this has been known for quite some time. Looking at some of the basic anecdotal evidence, some of the early research has been indicating this. So while certain microbes can have a negative impact on health, the truth is most of these are benefiting our body in a variety of ways. So most of the normal biota are good, and some of the major impacts they have that are well established at this point are influence on gut development. They've actually done studies on sterile animals where they take and remove all the bacteria from their gut during development, and then they look to see how that affects their development. And what they find is that the gut just doesn't really develop normally without microbes present. I haven't really seen any indications as to why that is, and I'm not sure if they know, but they've shown that the gut doesn't develop properly in animals. This is not done in people, thank God, right? We're not test rats, lab rats. But lab rats are, right? So they do this in rats and mice, and they find that when they remove the bacteria from the gut of mice and rats, they don't develop properly. So indication then is that this would assumably happen in people and other animals as well. So gut development then is influenced by normal biota. Lactobacilli are a major genus, or genera of bacteria, and same type of bacteria, by the way, they put in yogurt, one of the most common types, I should say. And what they find is that this has a major impact in the vaginal area of women, affects the pH and the acid-base content, and prevents the overgrowth of another natural microbe called Candida, a type of yeast, actually. So Candida is another type of normal biota, but only good in small concentrations. So if this microbe here gets too highly concentrated, it actually can cause a yeast infection. So an example, another place that microbes have a well-known benefit is to keep this balance in check between the bacteria and the yeast that occur naturally in the vagina. One of the biggest impacts is in the colon. This is where most of the microbes in our body exist, the large intestine, and they probably do a variety of things here, but the most traditional, it has been recognized as digesting our food or helping us digest our food. Now, the exact extent of this, I think, is still yet to be seen, but they've shown that they help break down food, they help us absorb nutrients, and different rates can take place here. So I'm going to try and put together a new online article talking about gut microbes and obesity, and they're finding that the exact makeup of microbes in the colon and exactly how they help you digest your food can actually make a big difference in how easy or how difficult it is for any individual to gain or lose weight. We all know there are some people who couldn't gain weight if their life depended on it, and then some people who can't lose weight if their life depended on it. And while there's probably a variety of factors here, including lifestyle and diet and genetics, the other big factor, as it turns out, is gut bacteria. And this may actually be the biggest factor. It's hard to say exactly. People are so diverse. There's probably not just one thing here, but once again, going back to studies in mice, they find that they can actually directly influence the body weight of a test animal simply by changing the gut bacteria and by keeping all other factors consistent in other terms of their activity, their food intake, calories, etc. Keeping all that consistent, they can actually trigger mice to be both obese or skinny by simply switching the types of gut bacteria and allowing that as the only variable to influence their natural body type. So in mice, you can see obese mice with one type of microbe or one series of microbes, and then skinny mice with another series of microbes. So research indicates that this has a really significant impact. But generally speaking, they're there to help us digest our food. And so then the extent of which they do that is going to make a difference in how easy it is for you to gain or lose weight. And the last thing here that's probably the most significant overall is that good microbes prevent the colonization of harmful microbes. Now, we're using good and harmful or good and bad as generic terms, but there are essentially good microbes that block bad microbes from establishing. This phenomenon is referred to as microbial antagonism. You want to recognize that term. It's going to be a part of the review here coming up. But basically, antagonism is a term that describes how good microbes block the invading microbes from establishing in the body. And this is an example where microbes can actually block the outside infiltration of microbes. They can also influence microbes that are already there, bad microbes in the body that are already there. But what they do this in two ways, I should say. One is they simply occupy body sites in the body that could be established by quote-unquote invading microbes or invaders. And essentially, there's only so much surface area of the body that a microbe could attach to. And by simply attaching good bacteria to those sites, there's no room left for bad bacteria to attach. So they simply block body sites and take up a presence there. And that presence then is preventative to other microbes from establishing. The other general way is that they actually release inhibiting chemicals. This kind of goes back to the idea of a selectivity of our body. And by releasing certain chemicals, they themselves act as almost like a selective presence to other microbes. So they can essentially kill off harmful invading microbes by releasing chemicals. So antagonism involves blocking and chemical inhibition. So this is where it's known, let me skip down to the bottom real quick. This is where it's known that antibiotics can have a major impact on our normal microbiota. Because we have good microbes that we need there, and because antibiotics tend to indiscriminately kill all microbes, what can happen is when we take antibiotics, we wipe out this balance of good bacteria. And when we wipe out that balance, it's almost like hitting the reset button on our body. And what we do is we kind of wipe that slate clean, and we allow these microbes to reestablish. And they're going to reestablish. You're never going to be able to keep your stomach sterile. Taking antibiotics won't remove bacteria permanently. But it will do is temporarily kill off a large number of what's there. And when you do that, then they're going to reestablish, and you can have a new balance of microbes. You may have more bad bacteria after that, or you may end up back with the same number of good bacteria. So there's no way to really know what's happening there. But the idea is that you have this balance of good microbes creating this antagonistic effect, and antibiotics can reset that effect. And the consequence is that you may reestablish the body with a higher number of harmful or disruptive types of microbes. So this goes along with the idea that normal biota really are only good in the correct numbers. So when we look at the overall balance, it works out that a microbe may only be quote-unquote good if it's in a small concentration. That same microbe may actually be bad if it becomes too numerous or too populous in the body. So adding to this idea of normal biota, good, bad, etc., you have to look at balance as part of that equation. It's not just a matter of what's there, but how much of it's there. So that's the idea here. So normal biota have to be balanced, and when they do occur in large numbers, that sometimes can be harmful. And this is a major impact of antibiotics. Antibiotics are life-saving drugs in certain cases, but they have a tendency to shift that balance and disrupt that, and that can sometimes, cause some pretty serious issues. One of the classic examples that I'm sure a lot of you are already familiar with, and a lot of you work in clinics and hospitals and probably know way more about this than I do, and that is Clostridium difficile, or C. diff. This is a spore-forming bacteria. I know we've talked about it, but this is one that it can cause serious issues after a dose of antibiotics, because you wipe out the normal biota and you wipe out that balance, and C. diff can thrive in the wake of an imbalance like that, and its numbers can go way up, and it can go on to disrupt the gut in a lot of really, really serious, harmful ways. So that's a classic example there of how microbes can reestablish after an antibiotic dose and go on to cause damage there. Now, what this next part is talking about is the acquisition of these normal biota, called resident biota here. Most microbes, when we come in contact with them, they may stick to our hands, they may stick to our body, but most of the time they're not going to stay long term. So every day we're exposed to microbes because they're ubiquitous, right? We talked about that. We get them on our skin, on our body, etc. In our food. Unfortunately, when we eat food, we're eating bacteria. We get some of those inside of our gut. But the truth is, our body is very, very good at keeping those from establishing, or in other words, from actually sticking permanently. So while we come in contact every day, most of these are removed or destroyed or inhibited by the body. Remember that our body is like a selective media due to its natural physiological conditions like pH and temperature, and then we also have microbes already there to prevent establishment of other microbes from coming in. So it's really, very difficult actually for microbes to come in contact with their body and actually stick, so to speak, or establish long term. So this has to do with the physiological conditions, the microbial antagonism, and then any microbes that get past those barriers then have to deal with the immune system in a lot of cases. And so that would make that even more difficult. So when we look at the acquisition of microbes, that it appears to be a small percentage of microbes that we come in contact that would actually establish in the body. However, this is more likely to happen early on during development. So what they've been studying for some time now is how the body begins its establishment with microbes from the time of birth and then going forward there. So what it looks like, based on the research, is that it looks like we are more or less sterile or close to sterile inside the placenta during development. Now, I honestly haven't looked up any new research on this. As I'm reading this here, I realize I haven't looked into this as much lately. But the traditional, I guess, assumption, or the traditional traditional information has stated that inside the fetus, or the fetus inside the placenta, is quote-unquote sterile. I'm not sure exactly. The textbook indicates that they're now contradicting this and that they think that this is not true. I honestly just haven't looked at some of the latest research here. If you're curious, you might look that up. But if we're not totally sterile inside the placenta, it's likely that we have a very small number of microbes there. So more or less, think about it, whether we're sterile or not, during development inside the placenta, inside the mother's womb, there's really very little microbes going on there. I think that's been established. There's not much going on there. So the first real events then that establish microbes in our body occur during birth. And it's actually the process called parturition, the technical term for giving birth. This is actually the major event in our life that establishes microbes in our bodies and on our bodies, on our skin. So whether or not we're sterile or not is a question that has yet to be answered, or at least maybe has, and I don't know. But the truth is that the biggest establishment occurs at the time of birth. Now, this has some impacts then. Real quick, we'll talk about some of the things that are happening here. They've done some interesting studies from the Human Microbiome Project showing that early on, the bacterial makeup actually changes in relation to some of the hormones released during different stages of pregnancy. So, for example, lactobacilli, which naturally occur in the vagina, can actually populate based on hormone triggers and actually increase in numbers, which is a good thing, because these are actually a good bacteria that we want to get, that the newborns actually want to be exposed to. So these actually become higher concentrated in relation to hormones during the pregnancy. Lactobacilli, one of the things that they do, one of many things, is they actually help digest milk, and they actually help provide a good stomach composition, a good intestinal composition. What I wanted to get to here, though, is the difference in the method by which a child is delivered. So you may be familiar with the difference between a vaginal and a cesarean birth, but these are the two ways in which a child can be delivered. One of these is considered natural. That would be the natural vaginal birth, in which the child passes through the vaginal canal. The other one would then be a cesarean birth, in which the child is artificially taken out through an incision made there in the stomach area. So the difference here may be minor in terms of how the child is actually brought into the world, but there appears to be a significant difference in terms of how the child is exposed to the natural biota of the mother. So a vaginal birth provides really what can be thought of as an inoculation of the child through the vaginal canal. This process here coats the child with a very thick layer of microbes, and that has a natural ability to establish these bacteria in and on the human body. When the child is delivered through a cesarean or C-section, the child bypasses this entire route and then bypasses all the natural coating and the inoculation that occurs with these types of bacteria here. So the question that's been brought up in recent years is, does this have an impact, and what's the significance here? And what they're finding is that it appears to have an impact. So when the child isn't delivered naturally through a vaginal birth, what can happen is that it doesn't get the natural microbes that you would find there in and on its body. And as a result, since our body won't stay sterile, what ends up happening is that it ends up getting these from an outside source. It ends up getting these from skin bacteria. It ends up getting bacteria from contact with the mother, contact with relatives, friends, hospital staff, etc. Anybody that comes in contact with it tends to then provide the major inoculation point for that child. So a vaginal birth appears to be the much more natural and appropriate way to inoculate the child, whereas the cesarean section leaves the child susceptible to inoculation through other events. So the question is, how much does it really matter? And since we know that C-sections are necessary sometimes, so I should clarify, these are necessary. There's medical reasons why a cesarean section needs to be done. There's sometimes where it's dangerous or risky for the child to pass through the vaginal canal, both for the mother or the child in some cases. So a C-section is something that is established as a necessary thing. Some questions have been brought up as to how many of these are actually necessary, and I'm certainly not capable of discussing that, and I don't know enough about it to weigh in on that too much, to be quite honest. But what I do know from research is that about 1 in 3 children in the U.S. are born through a cesarean section, and some of the indications from a medical necessity standpoint indicate that number is probably much too high. In other words, there's probably not that many children that need to be born for medical reasons in this way. And I think the speculation is that these are being done for convenience as opposed to medical necessity. There's a lot of convenience in a C-section. You get to pick the date, you get to pick the time, you get to kind of plan the birth as opposed to waiting for the child to come, which, it's risky. And there are some serious downsides to having a vaginal birth. Not to mention the pain and all that associated with it. But a C-section then is a good option for a lot of reasons. But one of the things that has to be weighed now with new indications of microbial impact is whether or not this has long-term impacts, and the early indications indicate that it probably does. So what they're showing is that there is a difference in the way the child is inoculated. Vaginal birth inoculates with one set of microbes, and a C-section tends to inoculate with a different set. And so you end up with a different microbial makeup in each scenario. And what the indication early on is indicating is that when a child's born through a cesarean section, that difference in microbiota can potentially lead to long-term impacts. And what they've shown is that kids born this way tend to have higher rates of allergies and also tend to have higher rates of childhood obesity. Now, whether that's a coincidence, whether or not this research is as solid as it should be, I honestly don't know. I haven't looked at these studies in close detail. So these are the indications, and as we know, sometimes these prove to be accurate, sometimes they don't. But it does appear that there's a significant link here, and it does appear that this should become a consideration during that parturition or child bearing process. So here's an interesting side note here. What they're now saying is that if you end up having a C-section, the way to kind of get around this whole thing may be really simple, a little bit gross, but you may just take some of the vaginal fluid and spread it on the child after giving birth. So perhaps the whole contradiction could be avoided simply by kind of doing an artificial inoculation, by taking some of those natural vaginal bacteria and kind of artificially inoculating the child after they've been given birth. Now, whether that will be as good, whether that's going to be as significant, I've not heard any indications on that, but I have read that this has been shown as a way to reestablish those bacteria, and at least indications are implying that this may actually be just as effective. So there's probably more on that on the web you could read about, but if you're curious. But that's some of the information there on the establishment of microbes. So we're born thought to be more or less sterile, or develop, I should say, in the womb sterile, and establish our first set of microbes, at least the most dominant microbes, at the time of birth. So consequences there, and more research as always to come from that. So I'd encourage you to look more up on that on the line. There's probably lots of information out there on that. So, all right, here we are, part two of 4. So go ahead, pause here. Again, I got lots of questions here, but only 4 parts this time. So. Okay, moving on to the next part here, we're going to talk about the nature of pathogens. So we've been talking more about the natural microbes and the normal biota and the relationships there. Next, we want to talk about the nature of the actual pathogen itself. Now, some of these terms here you've all seen. So this is all from Chapter 1, so I'm repeating this here to just refresh your memory on all this. What a pathogen is, it's just a microbe that has a negative interaction, type of symbiotic relationship gone wrong. And remember that there are, generally speaking, 2 types of pathogens, primary and opportunistic. So also recall, primary pathogens are those that can cause disease in someone with a healthy, normal immune function, so typically regardless of an immune status, whereas opportunistic pathogens cause disease in people with a compromised immune system. And also, I want to point out that opportunistic pathogens can also take advantage of location. In other words, they can establish infection in a body location that has a low immune status, areas where there's few immune cells to begin with. So opportunistic pathogens can happen in healthy people in body locations that are not heavily protected, such as the skin. This can take place sometimes in the urinary tract where there's fewer immune cells. So it's another aspect of opportunistic infection. This is a table from the textbook here, 13.3, showing other factors that affect the ability of an opportunistic pathogen here. So age, genetic defects, organ transplant, cancer, chemotherapy, etc. Now, when we look at a pathogen, there's a term that we use to describe the degree to which a microbe is pathogenic, and that's referred to as virulence. Really, virulence is another fancy word for pathogenicity, or the degree of pathogenicity. So if we say a microbe has a high virulence, that means that it's a strong pathogen. Okay, so virulence really just means the degree of pathogenicity. But you want to be familiar with that because that's a term you'll hear. So virulence of a microbe is something that can be objectively measured to a certain extent based on 2 general factors. So how pathogenic a microbe is, getting into the specifics of that, can be determined by 2 things. One is the ability to establish in the host, and I'm going to break down what that means here next. So establishment is a keyword that means a series of things. Generally speaking, it means attaching to the host, latching on and sticking to the host. So the ability to stick to the host, and then second, the ability to cause damage. So sticking and then any damage that comes after that are the 2 factors that determine virulence. Now the second one makes sense because if they cause damage, then it's understandable that they'd be considered a pathogen. But the first one is actually the most key here. It's actually the most significant. The ability to establish is the most significant factor in terms of pathogenicity. Okay, I'll talk about that in a second. Now, microbes can be also described in terms of what are called virulence factors. So this is any characteristic that contributes to the ability to cause infection. An example that we talked about already back in chapter 4 is a capsule. A capsule is that slimy layer that microbes produce that prevents phagocytosis, and that's an example of a virulence factor that makes them able to cause disease. But the truth is, establishment is really the most significant part of the virulence of a microbe. So, as it says here at the top, although the ability to cause disease is significant, it is imperative not to overlook the importance of establishment. And what I mean by that is that it's actually the case that lots of microbes could cause disease in the human body. The ability to damage human tissue is not something that's totally unique to primary pathogens and even opportunistic pathogens. There's a lot of microbes that could damage our body if given the opportunity. The truth is that they typically don't because they're not given the opportunity. They don't get the chance to cause disease. And the reason is because they have to first establish before they can cause disease. So establishment really is the fundamental process by which microbes have to enter the body first, attach to the body, and then the most difficult part is surviving the host defenses. So establishment involves all three of these things: getting in the body, attaching, and then the most difficult, surviving our host defenses, i.e. the immune system. So this is where a primary pathogen tends to have a heightened ability or a greater ability to establish, and that's really what makes them a primary pathogen. When we look at the technical characteristics that establish the difference between a primary and an opportunistic pathogen, establishment, use that word redundantly there, the characteristics that define a primary pathogen often have to do with the ability to establish. Okay, opportunistic pathogens then tend to be those that can cause damage but aren't so good at establishing, and only establish when they have an opportunity, which tends to be a lower immune system, and certain body locations can provide that opportunity, as I mentioned. So when we think about primary and opportunistic pathogens, think less about the disease they cause and the damage they cause, and think more about the ability to avoid the host immune system and the ability to actually stick to the host. That's really one of the fundamental differences between these two here. Honestly, when you look at the damage that they can cause, the damage is more or less the same. Given the full extent of primary and opportunistic pathogens, the average damage from all these different types, there's really no difference there. An opportunistic pathogen can wreck the human body just as much as a primary pathogen and vice versa. So the difference has to do whether they get that a chance or not, whether they ever have the opportunity to do that, which comes from establishment. So that's really important to understand. Now, when we look at how they establish, one of the first things we talk about is what are called portals of entry. And these are just a technical way of describing how microbes get in through different routes in the body, different routes of entry. So, for example, the body has skin and mucous membranes that can provide opportunity to get in. The gastrointestinal tract, anything that goes to the mouth and into our stomach. The respiratory tract, anything that goes into the lungs, or even the upper respiratory tract, the nasal cavity, or the urogenital tract. All of these represent openings, some more than others, but openings potentially in the body that allow the entry. And going back here just real quick, that would be, oopsie, this is why I don't like doing the bullet points. This would be the first step here in entering the host. So portals of entry provide the opportunity to enter. Now, these are all the common types here. I want to point out, and a question I might ask is, what's the most common portal of entry? And that is the respiratory tract. We tend to get sick most commonly through the lungs, tend to have a higher number of upper and lower respiratory infections. If you think back, that tends to be the most common way you get sick. The second most would be a gastrointestinal disease, stomach flu, stomach virus, stomach bug, so to speak. Those are the next most common. Now, when we look at a pathogen, whether it's primary or opportunistic, they can be somewhat defined as what route they typically enter through. So certain microbes, like the influenza virus as an example, they always enter through the respiratory tract. Some microbes can enter through multiples, but there's typically one characteristic route. Influenza comes to the respiratory tract. These are examples out of the textbook. Athlete's foot enters through cracks in the skin, so breaks in the skin and mucous membranes. skin, specifically for athlete's foot. But each microbe tends to have one or so routes that they're adapted to. Now, some microbes can actually have a variety of routes, and this increases the potential pathogenicity because it gives them more opportunities to establish. So, for example, Staphylococcus aureus, one that we've talked about, very, very notorious microbe, is able to actually enter through virtually any area of the body. Staph aureus is one of the most versatile pathogens that exists. It can cause virtually any kind of infection in almost any part of the body. There are exceptions to that, but it is very, very adapted. So it can come through the wound, the respiratory tract, the urinary tract infection, the gastrointestinal tract, and other areas as well. But these are the most well established. So it's such a high number of different locations it can enter through that it gives it that wide range of potential to cause infection. One of the reasons why staph infections are so potent. Another example of one I've mentioned before is Mycobacterium tuberculosis. This is the causative agent of tuberculosis. While it typically causes issues in the lungs, it actually can enter through the gastrointestinal tract or the lungs. So this gives it opportunities to get in the body through the two most common routes, either through ingestion or through inhalation. So while most microbes tend to have one route, some microbes, as these I mentioned here, can have a variety. Now, the next big thing in reference to establishment, so we're still talking about establishment, the next big thing is called the infectious dose, or the size of the inoculum. What this is talking about is how many microbes need to be present in order to actually establish an infection. In other words, what's the dose of microbes that you need to be exposed to before it's enough to actually do any damage? So what we have here is an inverse relationship in which if you have a small, what's called an infectious dose, that means a microbe is more virulent. And what that means is it takes fewer microbes to actually cause infection. And as a result, because then you would have to be exposed to less of them, there's a higher chance that you will actually be exposed. So microbes with a small infectious dose have a higher virulency, or considered more virulent. Microbes then with a large infectious dose are considered less virulent. And the idea is that you have to be exposed to a relatively high number of them before any damage actually occurs. So the truth is, we're probably exposed to a large number of potentially pathogenic microbes every day. In our food, for example, it's likely that we're ingesting things like Salmonella, E. coli bacteria that could cause infection of our body, but we rarely ever get a sufficient dose of those to actually do any damage. So we're getting a few cells here and there, and that's not enough to actually cause damage. So large infectious dose means less virulent, small infectious dose means more virulent. Now these are typically a range as opposed to an exact number, but generally speaking, it's a way to kind of characterize microbes that gives you an idea of what kind of exposure you need before they actually do any damage. Now, this can vary with the immune status. So people who have a low immunity, someone who's immunocompromised, would have a different relationship in terms of these numbers. So they're not a defined number, and they're not an exact thing. It's more of a concept that gives us an idea of how infection becomes established. So the idea is that you don't get establishment in most cases unless the infectious dose is met. Now that, again, can vary depending on your immune status, but that's the idea behind that there. Okay, so those are some of the things that relate to establishment. And again, establishment is the big factor in terms of how pathogenic a microbe is. Can they get into the body? Can they attach to the body? Can they avoid the immune system? We're actually going to talk about that next here. So I'm going to go on and continue this here. But go ahead and stop. Look at part 3. We got one more part to go. Almost done here. And we'll wrap this up. Okay, with the last section here, we're going to follow through and continue the logic behind the establishment process. So let's just back up real quickly here, and oopsie, going too far. Sorry. We're talking about establishment. So as I mentioned, this is a part of virulence, and establishment involves entering, attaching, and surviving the host defenses. So really, so far we've just talked about the entering, so portals of entry, and then the inoculation dose. Both of those relate to the first stage of establishment, okay? The next part here, we're going to talk about attachment, which is a little more straightforward. But really all we're trying to say here is that attachment is necessary. And this is done in different ways. But in most cases, a microbe only causes damage if it can stick or adhere to host cells and tissue. There's always exceptions to things like this, but generally speaking, this is a fairly common thing that has to happen. Here we see some different examples how this might take place. Bacteria, for example, can use capsules here, or Fimbriae here. Viruses use their spikes to adsorb to host cells. We talked about that in chapter 6. So one way or the other, attachment can take place, but it's typically a necessity for the establishment to take place. Now, one way the body attempts to reduce this is by flushing and secreting fluids and different chemicals that can be toxic to certain microbes. So this is where we go back to the idea of the body being kind of a selective media, and different microbes have a hard time establishing in different areas of the body based on the nature of the tissue, how they can attach. So for example, bacteria need some kind of mechanism to stick, and Fimbriae are a good way of doing that. Capsules are another way of doing that. So if they don't have that natural ability to stick, then they really don't have much of a chance. But even if they do have that, the body can inhibit that attachment through things like flushing and chemical inhibition. And remember that natural biota are already in these locations oftentimes. So these sites are already occupied, and the ability to attach as a foreign microbe can be very, very difficult. So lots of things make this process difficult, but the idea is that they need to attach, and that's usually a prerequisite for disease. So this can be something that often limits microbes, if they do get inside the body, the ability to actually attach and establish. From that standpoint, it can be pretty difficult. So that part's relatively straightforward. They need to get in, they need to attach, and there's just a couple of factors that relate to how they do that. The last step here in the establishment is surviving host defenses. And I'm not going to say a lot about that because we're going to talk about the entire immune system next. I shouldn't say the entire immune system, but the topic of the immune system and immunity is coming up next. So I'm going to save a lot of the detail for that. But for now, we just want to point out the fact that the last step of establishment involves avoiding the host immune system. And this is by far the most difficult part. So I stated that right here. So if a microbe gets in the body, if they attach, and if they're able to go through all of those different steps and all the different complexities that go along with that, the last step is typically going to still prevent them from establishing regardless, and that is our immune system. So our immune system is highly adapted to fight invading microbes and to keep our body in a natural balance. So one of the reasons why most of us don't get sick very often is not because we're not exposed, but because we have a healthy immune system to fight these off. So one of the first defending cell types are called the phagocytes. Phagocytes we talked about back in chapter 4, and these phagocytize invading microbes. So here's a figure showing what I was drawing on the board earlier. This is actually a real microscope image, artificially colored, but this actually shows a phagocyte, what it would actually look like digesting a yeast cell. So swallowing that up. So that's kind of a cool picture there. So one of the first lines of defense are phagocytes. We've talked about this, but to add a little bit more to that, one of the things that can make a microbe more virulent are what are called antiphagocytic factors. And these are characteristics, so these would be like an example of virulent factors that give them the ability to avoid phagocytosis. So we talked about the capsule, and slime layers can do this also, but really the emphasis of chapter 4 was a capsule. But technically both can do this, capsules really being the more primary mechanism. Another mechanism here, I skipped one, are a broad class of chemicals that microbes can produce called leukocidins. Leuko stands for white blood cell, or it stands for white, but it's in reference to white blood cell. So we'll talk about what are called leukocytes in the next chapter, and these are white blood cells. So leuko is in reference to a white blood cell, and then cidin is in reference to a toxin or a poison. And so a leukocidin is a chemical or poison that's toxic to white blood cells. So some bacteria produce these and can outright kill or inhibit white blood cells. And that would be most definitely a characteristic of a primary pathogen. And this is one way that they can get around the immune system. So primary pathogens can produce leukocytins. They can have capsules, sometimes produce slime layers. I keep doing that to suppress the immune system. So regardless of whether you're healthy or not, these characteristics can actually contribute to infection regardless. And then the last characteristic here, which is by far the least common, is a trait of certain microbes, such as the one that causes tuberculosis, Mycobacterium tuberculosis. And they can actually survive inside of the phagocyte. So going back here real quickly, if the microbe, in this case, is ingested or phagocytized, what can happen is it can actually survive inside of this little capsule here called the lysosome. It's actually called the phagolysosome. And bacteria like Mycobacterium tuberculosis will actually not only survive inside of this phagolysosome, in other words, after they've been phagocytized, they not only survive in this, but they can actually replicate within this and actually grow within the white blood cell. And they can actually duplicate themselves, make copies of themselves, and then break open the cell and then go on to spread to other parts of the body. So microbes that have any of these characteristics here can have a much easier time avoiding the immune system because of the fact that the immune protective factors are less effective. So again, these are characteristics typically of primary pathogens that allow them to avoid the immune system. This is just a relatively short list of some basic factors and not the full extent of primary pathogenic factors, but gives you an idea of some of the things that we're talking about when we talk about primary versus opportunistic pathogens. So to clarify, opportunistic pathogens typically don't produce any of these sorts of characteristics and would only then be able to establish given the absence of a phagocytic response or a white blood cell response, or in a location of the body where there's fewer white blood cells. So for primary pathogens, this is how they avoid those when they're present. Now, the last part here, we're going to talk about the disease process itself. So once a microbe is established, it can start to populate and start to grow to large numbers. And the high population of a harmful microbe then can lead to damage of body tissue. So the way this generally works is of one of two ways. And usually it's a combination of both, almost always a combination of both. But, excuse me. One way is through the direct release of enzymes and toxins. You kind of just think of these as toxins in general. They're technically different, but they release harmful substances. And one of the things that makes microbes harmful is the fact that they can release toxic substances. So this is considered direct damage. They release a substance that directly affects the body, and this is considered a form of direct damage here. Now, so that's one way that they can cause disease. The other way that they do it is through indirect damage. And what happens here is that their presence triggers an immune response by the body, which is what you want to have, typically anyway, inflammation. But it's actually the inflammatory response that ends up causing damage. And so in some cases, we have a situation where a microbe simply being present can trigger an immune response that makes the process worse. So your immune system trying to actually help ends up causing damage. So as an example, what I already mentioned was psoriasis. Psoriasis is an example of where the symptoms are actually caused by the immune system. Now, the exact underlying cause of psoriasis is still being just determined, but the idea is that there's something happening there where the immune system thinks that it needs to attack. It thinks there's a problem that it needs to go handle, and as a result, it creates an inflammatory response. You get redness, swelling, itching, irritation, sometimes damage from that, and it ends up causing more harm than good. And sometimes there's a balance where you have a little bit of harm but more good, and sometimes you have more harm and less good. So there's a range here. It's not always good or bad, but inflammation in itself can be highly damaging. And in some cases, that's the primary cause of damage. In other cases, the primary cause is the direct damage from enzymes and toxins. Most of the time, it's a combination of both, but we'll talk about examples of these here. So microbes like bacteria are especially good at releasing extracellular enzymes. These are enzymes that they secrete outside of their body. These are sometimes called exoenzymes. And they're typically molecules that microbes release to digest food in their environment. They do this in the natural environment. Fungi do this also. Protozoa can do this to a certain extent, but mostly it's fungi and bacteria. And they release enzymes to help break things down. They're digestive enzymes, so they digest a substrate. So they can do this in the natural environment. And a microbe that lives in the soil, for example, can do this to food in the environment. But it can also do it inside the human body, and they can use the human body as a food source, much like they would use, say, a rotting tree as a food source in the natural environment. So the difference is what kind of digestive enzymes they produce. So when we have a pathogen, one of the things that it can do is literally use the host tissues as a food source. And it does this by releasing digestive enzymes, so to speak, or toxins that break down the body and release nutrients for the microbe to use as a food source. So we see this in bloodborne pathogens, for example, and they can release enzymes that can break down red blood cells, as an example. So they use this to get nutrients. They can also do this to help them spread. So they can break down some of the tissue barriers, and this can actually allow them to move throughout different areas of the body. So some examples of enzymes that help microbes cause damage are things like mucinase. Mucinase is an enzyme that helps break down the mucus coating found around the mucous membranes of different body locations. Show a quick figure here from the web. Mucous membranes can coat a variety of different areas of the body, such as the upper respiratory and the lower respiratory tract, and they're basically just a layer of slime that the body produces to help trap microbes and protect the tissue beneath it. So most of the time this is effective. Most microbes get stuck in this mucus layer and ultimately get kind of inhibited by it. But certain microbes can simply digest this layer and move right through it, and that's the role of an enzyme like mucinase. Keratinase, collagenase, these are both proteins in the skin that give skin kind of its protective characteristic, give it kind of a strong and flexible characteristic. Another one is called fibrinogenase, or fibrinogen, excuse me, another common protein. But microbes can break down these by releasing enzymes such as keratinase, collagenase. These are enzymes that break down those types of proteins. Another one called hyaluronidase breaks down a chemical called hyaluronic acid, sometimes referred to as cellular glue. It's a substance that kind of glues, so to speak, animal cells together, gives them kind of a coherent type characteristic. It keeps tissues kind of somewhat loosely connected. So all of these give the microbe the ability to break down human tissue or different components of it and use that as nutrients, or simply move through it so that it doesn't act as a barrier as it normally does. And by the way, I want to point out here, enzymes are typically named in a way that they end with the suffix "ase." So when you see ASE in a word like this, that's an indication that it's an enzyme. So the suffix is usually "ase," and then the prefix most of the time has to do with what the enzyme is doing, although that's unfortunately not as consistent as it should be. But mucinase, for example, is in reference to mucus, and then the ase indicates that it's an enzyme. So keratin, ase, etc. So that's a way to recognize what the word is indicating, enzyme, and then what effect it has typically. So these are all examples of enzymes that are released into the body that can break down different body tissues. Now, enzymes are similar to toxins, but technically a little bit different. The difference between a toxin and an enzyme is often indistinguishable from the effect that it has. And in fact, a lot of times the terms are used interchangeably, even though that may not be accurate. Technically, an enzyme is a molecule that catalyzes a biochemical reaction. It breaks things down in a way that serves a role in metabolism. Toxins tend to be a chemical that interferes with a physiological and metabolic process. So they're technically different, but the effect can be indistinguishable. So a lot of times we just refer to them as toxins, even though there's technically a difference. Okay, but I'm not going to try and catch you up on the differences here. I don't really worry about it too much. I'm just trying to point out that there's technically a difference here. even though really even times doctors and physicians use the terms interchangeably. So it's one of those things that isn't real important to know the difference, but just recognize there is actually a difference. So toxins tend to block normal processes. So for example, a neurotoxin can block the activity of neurons. So here's another figure I pulled off the web, showing on the left here a normal neuron sending signals across the synapse and communicating with another cell here by releasing these little neurotransmitters. A toxin like this one here, called the tethered toxin, can bind and block the activity of the enzyme, or excuse me, of the neuron, and block the normal response that this neuron has, the normal communication that it has. So this is an example of how a toxin might work. It actually physically adheres and blocks the normal functioning of a physiological process. So that's the technical difference here. I misspoke there, by the way. This isn't called a tethered toxin. This is showing a toxin that's tethered to the receptor, meaning that it's attached to it. That's what that means. Okay, so examples would be like neurotoxins. Enterotoxins are those that act on the intestines. Hemo means blood. These are those that affect red blood cells. And then nephro, or the nephrons, is in reference to the kidneys, and these are toxins that affect the kidneys. So once again, these are just a short list of examples from the textbook. The whole list of toxins would be in the thousands. So these are, by the way, these are classes of toxins. I should also mention these are not specific toxins. These are general classes of toxins based on what body locations they affect. So for example, there could be tens if not hundreds of neurotoxins, and the same for each of these here. Not all of them have quite as many, but there are different types of specific neurotoxins, for example. So we're just defining them as kind of a broad category of toxin here. When you look here, this is a figure from the text showing how some of these toxins and enzymes can play a role. So here's a figure showing bacteria in green, and some of the enzymes they release as these little black dots. And what this is showing is how they can break down. This would be the hyaluronic acid connecting cells together, this cell glue, and this allows them to actually move through the tissue and invade into deeper regions of the body. Here we see some exotoxins breaking down the outer layer of epithelial cells. So again, the difference here can be almost indistinguishable. Even though these technically have a different mechanism of action, the effect can be almost identical in terms of what's happening. So that's why the terms are so interchangeably used. So the last part here I want to mention with the last slide is that viruses have a very different effect on the body compared to bacteria. And they typically don't release enzymes and toxins. And what they typically do is just infect the entire cell and damage it from within. And we've talked about viruses already, so you have an idea of kind of what they're doing. But generally speaking, they cause internal damage to the cell from within in a very different manner. And really the noticeable symptoms that they trigger are all part of the indirect damage in which the body is responding to the virus. So going back here just real quick, we talked about two different ways that they end up causing damage, and that is through either direct damage, enzymes and toxins, or indirect damage from the inflammatory response. And while viruses certainly do damage cells internally and somewhat directly, a lot of times it's the symptoms that come from our immune system that actually have the greatest impact on our body. So when we get sick from a viral infection, we start getting the fever, the headache, the swelling, the temperature, and all of that, vomiting, etc. Almost all of that is a direct response from the immune system. And it's not very likely that you would be feeling the direct damage from the viruses. Now, that may differ if you have a really serious viral infection. If you have a life-threatening viral infection that goes on for an extended period of time, then you can certainly get into situations where the microbe can affect your ability to breathe, or it can affect your ability to move fluid throughout the body. So if the extent of the damage goes on long enough, then you can get a really serious impact there. But generally speaking, the effect that they have is that they cause cell damage and trigger an immune response. And then the immune response is what ultimately leads to all the symptoms that make us temporarily miserable. So this is really more of an example of indirect damage or indirect triggering of symptoms as opposed to the direct damage caused by enzymes and toxins. So splitting hairs a little bit here. Viruses, to clarify, do actually cause direct damage, but typically it's the symptoms and our awareness of the illness, rather, that comes from our own body's immune response, and less so of the actual damage from viruses themselves. So most viral infections are relatively harmless, as it says down here, as long as the immune system is able to remove them. So typically, they cause a little bit of damage, they trigger the immune system, it kicks in, and that's when we get the symptoms. And usually the symptoms go away when the immune system is done handling the infection, and we recover in the wake of that. So a little bit of difference there in terms of how the damage is caused. And just to clarify, bacteria typically do both. They typically are going to cause direct damage through the release of toxins and enzymes, and then they're also going to trigger an inflammatory response, typically at the same time. So usually the consequence of a bacterial infection is a direct and indirect consequence. So, okay, here we are at the end of this chapter 13. So review part 4, that's the last part, no part 5 for this one. So, all right, go ahead and look at these review questions. And as always, let me know if you have any questions in class or email me. And I will see you next week in class. So have a great week. Goodbye.